next up, as we move him over, is Dr. Joe Abdo, the CEO of Stella Diagnostics. And I believe this is an OTC ticker, JMDP. JMDP. I will double check that as Joe comes over. Joe, did I get that right?
Yep, that's it.
Awesome. That is the one. Well, wonderful, man. Welcome. I love the mountains behind you. Where are you presenting from today?
Salt Lake City, Utah.
Awesome. The Great Salt Lake. All right, my friend, I'll let you get to it.
All right. Thanks so much for having me. And this has been a great morning watching the companies. I was able to catch a couple companies present this morning. And I'm so excited about all the great work being done in the molecular oncology fields. Hats off to all the great small cap companies that are doing landmark research and commercialization of clinical products. So I'm just really thrilled to be here, and thanks for having me. Good afternoon. Again, my name is Joe Abdo. I'm scientist, co-founder, and CEO of Stella Diagnostics. Obviously, as I said before, we're really honored to be here at the Benzinga Small Cap Biotech Conference. And we're also excited to show you about how our leading edge technology is designed to improve the standard of care for over 60 million Americans living with acute or chronic esophageal disease. This presentation today will demonstrate how our tests can notify physicians if their patient's tissue is stable, progressing, or turning cancerous. Here are our forward-looking statements, which can be found on our website, www.stelladx.com. So Stella Diagnostics is currently based in Salt Lake City, but we are a pan-national organization operating multiple functions coast to coast. Stella DX uses targeted mass spec proteomics, and we believe that informing physicians of the expression patterns playing a role in the pathogenesis of GERD, Barrett's esophagus, and esophageal cancer early in the treatment plan may extend or even save lives. A subtype of esophageal cancer called esophageal adenocarcinoma, which will herein be referred to as EAC, is a cancer of great public importance because of rapid increase in incidence and poor survival outcomes. The cancer arises from a premalignant condition called Barrett's esophagus, which is a complication of gastroesophageal reflux disease, commonly known as GERD. Chronic injury to the esophagus because of acid reflux initiates a metaplastic change in the bottom of the esophagus, resulting in the conversion of normal epithelium to a premalignant dysplastic surface. Patients diagnosed with Barrett's have a 100-fold higher risk of developing esophageal cancer compared to the general population. The reasons for poor survival are twofold: late diagnosis and limited treatment options, with a specific deficit of biology-guided therapies. Esophageal adenocarcinoma is the deadliest form of cancer relative to incidence in the United States, but is also one of the least understood or publicized malignancies. Indeed, EAC is the fastest rising cancer in North America, with a 600% increase over the past 25 years. 4 out of 5 esophageal cancer patients present to their oncologist in the advanced stages of cancer progression, which is why the average overall survival duration is only 13 months from the day of diagnosis. Stella Diagnostics aims to make that percentage of late-stage presentation significantly lower with a multi-pronged diagnostic approach so that high-risk patients will be identified earlier when the disease is still in a curable state. Every 30 minutes, someone in the United States dies from esophageal cancer, in part due to inefficient screening. The current pathology standard of prognosis for these diseases is gross and microscopic inspection of esophageal biopsies. Patients diagnosed with Barrett's esophagus receive endoscopic evaluation, routinely ranging from every 3 months to every 3 years, depending on severity. During these visits, the esophagus is observed with a camera by the treating GI, and biopsies retrieved during this procedure are stained by in-house pathologists, and aspects like cell structure and organization, histology, and inflammation are noted and reported back to the GI. Very little, if any, molecular information is assessed with current standard screening protocols for this pre-malignant condition. Stella can provide robust molecular information using just a tiny bit of leftover biopsy tissue taken during these procedures, which means that we can seamlessly provide more information on the presence of disease drivers in Barrett's tissue without disrupting the standard screening workflow. Stella has discovered the expression trends of novel biomarkers that illuminate the carcinogenic proteomic environment that leads to an increase in cellular proliferation, invasion, migration, and cell survival, as well as chemotherapeutic resistance to the most commonly prescribed drugs for esophageal cancer. The presence and activity of the biomarkers in our panel may provide a colorful picture to physicians, showing if their patient's tissue is behaving normally or beginning to show hallmarks of pathogenesis that sometimes go undetected with traditional diagnostic approaches. Stella's first aim is to offer physicians insight into molecular properties of patients' esophageal tissue to reliably determine if the disease is stable, progressing, or turning cancerous. Secondly, if a patient has already developed cancer, we will have a panel that assesses proteomic expression patterns associated with increased benefit or resistance to all the FDA-approved therapies for esophageal adenocarcinoma. Aim 3 requires additional R&D to assess if our method of discovery of novel markers will be applicable to tens of millions of GERD patients and evaluate if inhibition of our novel markers can arrest the development of Barrett's. Stella Diagnostics aims to be a one-stop shop for in-depth molecular analysis for the entire progression cascade of esophageal disease. Here is a representation of 4 markers in our panel, where clear overexpression was observed as esophageal tissue progressed from normal to Barrett's, which is precancerous, and then on to cancer. Our bioinformatics algorithm elucidated these markers that were upregulated in this fashion, filtering by their roles in carcinogenic processes that are well described in the literature. The majority of these proteins are viable drug targets with proper binding sites, which could lead to therapeutic programs down the road. Stella's diagnostics assays utilize mass spectrometry to offer high throughput, multiplexable, and confident data, assessing multiple disease-specific biomarkers simultaneously. To assure highly accurate quantification, we spike in a known amount of our synthesized markers representing the proteins in our panel. The markers are isotopically labeled so we can detect them with ease and subtract the amount of protein spiked in from the endogenous signal yielded by the patient's tissue. This gives us extremely accurate and precise quantification of all biomarkers concurrently. The results of the expression trends of the entire panel will translate into a risk score, notifying physicians if the biopsy represents low, intermediate, or high risk for disease progression. Our Stella 102 panel consists of proteomic biomarkers affiliated with improved response to targeted therapies represented by green boxes, or chemotherapy shown in red, as well as resistance to various chemotherapeutic agents that are FDA approved for esophageal cancer. Knowing the expression status of this panel could assist oncologists in optimizing first-line patient management strategies for EAC patients by notifying which targets are present to increase efficacy or which resistance markers are expressed that would inhibit the anti-cancer effects of the associated drug. The crux of our technology relies on the most superior platform for detection and quantification of human proteomics known as mass spectrometry. Utilizing this method allows Stella to deliver accurate results void of subjective interpretation and false positives from a very small piece of fresh or fixed tissue in a timely manner. The runtime is about 45 minutes on the mass spec, and all markers are detected and quantified simultaneously. Stella's tests require only 8 millimeters squared of tissue, and we can deliver the results to the ordering physician within 1 to 2 weeks of clinical biopsy retrieval. The novel markers we discovered may be the most consistently expressed proto-oncogenes in EAC. The proteins in our proprietary panel have extraordinary and consistent overexpression in esophageal adenocarcinoma tissues. Compared to the 8 most scrutinized markers for gastroesophageal cancers, which are currently offered by various molecular diagnostic companies, Stella markers with an overexpression range of 85 to 100% in EAC tissue possesses unprecedented diagnostic utility, which should call for routine inclusion in pathological analyses and clinical patient management strategies. For example, HER2 targeted therapy, such as trastuzumab, is the only FDA-approved targeted therapy for EAC, yet is only overexpressed in 18% of EAC tumors. Therefore, trastuzumab would be ineffective in 82% of EAC cases. The high prevalence of overexpression of our newly discovered markers and their potential druggability may yield exciting anti-cancer therapy projects in the future. Switching gears to the commercial side of Stella, we have a bird's eye view of the potential market size for oncological prediction or therapeutic optimization of esophageal disease. 1 in 4 adults, or 65 million Americans, have GERD, of which about 6 million patients develop the chronic esophageal disease known as Barrett's esophagus. Around 20,000 new cases of EAC are diagnosed each year that are developed from Barrett's. About 1 million patients with the lowest grade of Barrett's, known as metaplasia, are seen by a GI for upper endoscopy once a year. About 650,000 upper endoscopies are performed on patients with high or low grade dysplasia every year. This equates to a total testing volume of 1.6 million upper endoscopies for Barrett's patients per year. And to our knowledge, the average molecular diagnostic test in the oncology space costs roughly $1,900, making our total addressable market around $3 billion per year, and will continue to rise sharply along with the increased incident trends of lower esophageal disease in the United States. There are 2 other companies in the molecular diagnostic space for prediction of Barrett's esophagus progression. Company A looks at genomics to quantify the mutational load of DNA residing in diseased tissue. Mutational load is only one component that may be predictive of disease progression. Additionally, no actionable data can be retrieved from this test because, very simply, therapies target proteins, not genes. Company B and Stella both examine the proteome, which consists of real-time proto-oncogenic markers and disease drivers in the patient's tissue. The main difference between Stella and Company B is Stella's mass spec technology is antibody-free, which avoids issues such as nonspecific binding and subjective interpretation. Mass spec provides a more reliable and accurate test for physician decision-making, which we will pair with our penetration pricing model. Stella's fast turnaround time helps clinicians make their treatment decisions more quickly, which can have a direct effect on patient outcomes. And lastly, Stella Diagnostic panels require the least amount of tissue, allowing for the majority of the precious biopsies to remain available in-house for internal standard of care pathological inspection. Ultimately, Stella's goal is to make supplemental diagnostic processes easier, cheaper, faster, and more reliable for everyone involved, while delivering the most actionable molecular information to the providers. The last 6 months have been very busy for our young company. We have received tremendous support from the investment and clinical communities and are well on our way to being able to offer multiple product candidates for GI specialists. Company-wise, we have incorporated and became a public via reverse merger with JMD Properties. We have secured funding for company infrastructure, assay development, validation and optimization, and talent acquisition. Our provisional patent was accepted by the U.S. Patent Office, with a non-provisional patent prosecution commencing in Q3 of this year. Clinically, we have aligned with world-class medical institutions such as the Mayo Clinic and the University of Kansas Medical Center for prospective and retrospective studies of our assays' predictability and sensitivity. The Stella 101 diagnostic test is built and validated for targeted mass spec and will continue to be optimized via ongoing multi-institutional studies. We have presented data at global oncology conferences, and our work has been published in the European Journal of Cancer and in other publications under review at the Journal of Gastrointestinal Oncology. We were honored to receive the 2020 BuzzABio Award by the Biotechnology Innovation Organization for our breakthrough technology and team of domain experts developing our product candidates. Lastly, our stock ticker symbol change to STLA has been submitted to FINRA and is pending. A listing of our up-to-date achievements and milestones can be found on our website. This quarter, we have had consistent news flow covering our landmark clinical research partnerships and recruitment of key board members with decades of experience of asset management, investment banking, and corporate development. Our collaborations and achievements have been published in the Wall Street Journal, Genome Web, Precision Oncology News, Yahoo Finance, and many other outlets. We expect to maintain this consistent cadence of press coverage and accomplishments for the remainder of the year. Here we have a 30,000-foot view of the upcoming company milestones for our next 4 quarters, which include finalizing data analysis of multi-institutional studies, and assessing clinical utility of the Stella 101 assay. With these results, we will finish a non-provisional patent prosecution, present data at conferences, engage key opinion leaders, continue robust R&D, including assessing our assay's compatibility with less invasive esophageal tissue retrieval techniques, perform prospective studies, develop and validate our second product candidate, build or partner with a centralized CAP-accredited and CLIA-certified laboratory, ultimately leading towards commercialization. The method of discovery and subsequent clinical utility were designed and elucidated by domain experts and practicing physicians in the fields of clinical science, surgical oncology, basic science, molecular oncology, and proteomics, with a combined 70 years of experience and 450 peer-reviewed publications, respectively. We assembled a complete team able to identify clinical limitations in the screening and treatment of Barrett's and EAC, and design studies to discover novel markers for high throughput molecular scrutiny for precancerous esophageal disease. Thank you so much for attending this presentation. We're thrilled to be here with all of you at Benzinga Biotech 2021. We hope you're as excited about our clinical offerings as we are. If you have any more questions, please contact me, Joe Abdo, the CEO of Stella Diagnostics, or check our website, stelladx.com. And thanks again for having us. We'll now open up the session for questions.
Thank you, Joe. Thank you very much. I have a couple I'm just going to rattle through if that's okay with you.
Sure.
So we have, first of all, the ticker change, STLA. You've got it in with FINRA. Any timeline on that in terms of when that's going through? And is that still on OTC or is there an uplisting there?
Not an uplisting yet, but it is in our plans within the next, I would say, 4 to 6 quarters.
Okay.
But the STLA ticker change, I wish I had that answer for you. We check pretty much every other day, and it's still pending. But I'm expecting it probably by the end of Q2, beginning of Q3, which would be great.
Yeah. Well, listen, man. The only thing slower than the biotech clinical process are the regulatory agencies. So
I didn't want to say it, but I appreciate.
I will. I'm here for you. Okay. So a couple more from the chat here. The biopsies themselves, right? I just want some clarity for this investor who's asking, where are these biopsies sent? Are you guys doing the work in-house? Are you sending them off to other labs like a LabCorp or a Quest or something like that? Or are you doing this work yourself?
Yeah, we're doing the work ourselves. So and I get this question a lot when speaking with investors. A lot of them will see this presentation and ask if this is a test that's available at the bedside. And we work as a centralized laboratory, kind of like, Foundation Medicine and companies like that, where, we have a CAP-accredited, CLIA-certified lab that we're working with currently. We will at some point build our own brick and mortar CAP-accredited, CLIA-certified lab. But we'll do all of that stuff in-house. And the nice part about our clinical workflow is that the biopsy gets taken by the GIs. They send that down to their in-house pathologists. And all we do is ask for a test requisition form. And we just require 2 slices of that biopsy that's typically left over after they do their in-house pathology. And they send it to us at our centralized lab, which is currently located in Rockville, Maryland. And we do our testing there. Great.
So one final note to close on here, and we have about 30 seconds left, but I think a good note to leave our audience with here. What is it about these novel biomarkers that you guys are focusing in on and the data that you're collecting that makes Stella Diagnostics uniquely qualified to be informing these physicians, to be helping them improve their diagnostic processes and to obviously improve patients' lives?
Yeah, great question. When I was working with my clinical and academic collaborators, we made these discoveries. We designed this, beautiful, what I call a molecular dragnet using discovery mass spec, not using targeted mass spec like we use at Stella Diagnostic, but discovery setting, which is we didn't know what we were looking for. We found about 20 biomarkers that we thought were playing a role in the pathogenesis of this esophageal disease. And the majority of them we typed into PubMed using every combination possible, and there is no link to these biomarkers with esophageal disease or progression. It has been well described in other cancers as disease progression, but nothing in esophageal disease. So the only people that know about these markers are us. And that's why we're going through an arduous patent process right now so that we can get this novel information protected, and then we can share it with the world. And we want to be able to list all of the information about these unique biomarkers so that we can, better the entire field of GI screening and therapy. And so, we're just waiting on that. And then we'll be able to share these unique novel biomarkers that are currently undiscovered by, the clinical science community in general.
Awesome. Well, thank you. Thank you very much again, man. I really appreciate it. Dr. Joe Abdo, Stella Diagnostics, JMDP. I'll get it one of these days.
That's all good. Thanks so much for having me. This was awesome.
Thanks, Joe. Take care, man.